Good afternoon, children. In the last class, actually, we were discussing about the packaging of DNA helix. In that one, we were discussed about the two important topics. That is nothing but the packaging of DNA in prokaryotes as well as the packaging of DNA in eukaryotes. Also, the structure of nucleosome, isn't it? So these are all things we were discussed in the last class. In this class, we will start the discussion with respect to the another important topic. So this is very important, especially in the field of genetics. Okay, the search for genetic material. Okay, so the search for genetic material. So there are three important experiments we will discuss under this topic. In that one, first one is transforming principle, which is conducted by Frederick Griffith, and there is another important experiment, the biochemical characterization of the transforming principle. Also, the third one is Hershey's experiment. It is popularly called Blender's experiments. So these three important experiments today we will discuss. Okay. So let us discuss one by one the major intention of all these three experiments. So to prove which is the genetic material. Okay. Yeah. And you know very well that in the most of the organism, the DNA is the genetic material that controls hereditary characters, isn't it? Okay. See, let us discuss the first experiment in the search for genetic material. The first one is the transforming principle. Okay. So the transforming principle was discovered by Frederick Griffith in 1928. Okay, please note down there are few points it is not present in your NCERT, so those are all very important for the purpose of your competitive. Okay, the transforming principle experiment was discovered by Frederick Griffith in 1928. Okay, and the transformation, what is a transformation? Here the question arises, what is transformation? The transformation is a kind of sexual reproduction in bacteria, okay, where the genetic material of donor bacteria transferred into recipient bacteria without physical contact. Okay, so this is called transformation. I will repeat once again, what is the meaning of transformation? The transformation is a kind of sexual reproduction in bacteria where genetic material of donor bacteria transform into recipient bacteria without physical contact. Okay, yeah. And see, his experiment, actually it is purely based on Streptococcus pneumoniae. Okay, so that means the Frederick Griffith selected for his experiment, so that means the Streptococcus pneumoniae. So it is generally causes pneumonia in human beings. So that is a kind of a respiratory disorder. You must have heard about it. Okay. So then, see here, whatever he selected there are two strains. Okay, for his experiment, as I said, the Streptococcus pneumoniae only, but so there are two strains he selected. Okay, so that means Streptococcus pneumoniae bacteria such as smooth shiny colonies, he called them as a S strains. Okay, they are also virulent. Okay, the poisonous. That means, so how he selected based on the smooth shiny colonies. Okay, and they are virulent, they causes the disease. Okay, that means they causes the pneumonia in organism, which is having generally a polysaccharide mucus coat, causes pneumonia. And he selected the another one, Gruff colonies. They are commonly called R strains. R strains are generally they are non-virulent or non-poisonous. Okay, no mucus coat is present. In case of all strains, that means mucus coat is absent. They do not contain, okay, the mucus coat, as well as they do not causes pneumonia in organisms. Okay, so this is how he selected the Streptococcus pneumoniae two strains. There is a smooth strains and rough strains, or we can also say smooth virulent strain, rough non-virulent strain. Okay, so this is how he selected. Then see, after the selection of these two strains, two different strains, the H strain bacteria have a mucus, so that means polysaccharide coat and called capsulated bacteria. Okay, and the R strain does not have mucus coat, and they are called encapsulated. That means capsule is absent. Okay, and so whenever he started the experimentation, he conducted majorly four type of the experimentation, and also he observed. Okay, see, when he started the experimentation, the S strain is virulent. That means he is identified that S strain is poisonous, that means virulent, and caused pneumonia in mice. When he injected to the mice, what happens? The S strain is virulent and cause pneumonia in mice and dies when it is get infected. Okay, so that was the first experiment, or first possible observed experiment. Then the second one, R strain. Okay, R strain is a non-virulent and does not causes pneumonia in mice when even it is injected. Okay, whenever it is injected to the mice also. So the R strain is non-virulent and does not causes pneumonia in mice when it is infected. So these are first two observations, okay, he made and he completely noted. Okay, and in third experiment what he made, he killed S strains. That means he given the heat treatment to the S strains in non-virulent and does not causes the pneumonia. Okay, after the giving of heat, the heat treatment to the S strains, he injected, or he gripped, infected the mice with the heat killed S strains, so that the mice were not died. Okay, that means does not causes pneumonia. Okay, so these are the three observations he made in the beginning. And in fourth case, what he made actually, the heat killed S strains, that is smooth strains, he mixed with the live R strains, injected into the mice. The mice now developed the pneumonia and dies. So that means the mixture of the heat killed S strains with live R strains, whenever it is injected to the mice, what happened in the fourth case? The mice developed pneumonia and dies because of pneumonia. Okay, so these are the four observations he made. Okay, these are the four observations as well as the four possible experiments he conducted. Okay, see, as I said, he on the basis of these four experiments, what he discovered, he recovered live S strain bacteria from the dead mice, as well as Austrian bacteria had shown the genetic material. So that he recovered, what he recovered? He recovered live smooth strain bacteria from the dead mice. Okay, and rough strain bacteria had shown the genetic material has been transformed by the heat killed S strain. Okay, so that bacteria to the Austrian bacteria. So some transforming principle. So that means from the above experiment, what he observed, some transforming principle transferred from heat-killed S strains bacteria, so that had enabled R strain bacteria to synthesize smooth polysaccharide. Okay, so that whenever they develop the smooth polysaccharide coat and they become virulent S strain. Okay, so this is how actually the transformation of the R strain to the S strain is due to the transfer of genetic material. Are you getting what I am telling? So that means what he concluded from the above experiment. So that means the feeder graft, he concluded that some transforming principle transformed from heat-killed S strains to R strains, so that it enabled the R strains to synthesize smooth polysaccharide coat and become virulent. Okay, so this is due to the transfer of genetic material. Okay, so this is the conclusion he made by his transforming principle experiment. However, so from this experiment, the biochemical nature of genetic material was not identified. Okay, so because the question arises, he said that it is only a genetic material, but he is not told clearly. So that means, however, the biochemical nature of the material was not identified from his experiments. Why? The question arises whether it is a protein or RNA or DNA. So these answers are not given, or he is not able to give the justification of this. Okay, so that means whether whatever the genetic material is transformed, that is from R strains to the S strain, whether it is a protein or RNA or DNA. Okay, so this is the drawback of the transforming principle was given by Frederick Griffith. Remember, sometimes they will ask one last question: what are the drawbacks of transforming principle? Then you should write. So the biochemical nature of the genetic material was not identified from the transforming principle experiment, because the question arises whether it is a protein or RNA or DNA. Okay, so this is about the first experiment with respect to the search of genetic material. Okay, fine. The next experiment, so there is another wonderful experiment, the biochemical characterization of transforming principle. Okay, so the biochemical nature of transforming principle was discovered by, there are three names of the scientists: Asper Avery, Colin MacLeod and McCarthy. Okay, so these experiments, they were conducted between 1933 to between 1944. It's about 11 years. Okay, it's about 11 years. Remember, they will ask sometimes the duration of the experiments also. Okay, yeah. See here, the prior to their work, genetic material was thought to be the protein. Okay, so as I said, that means these three scientists, that means Oscar Avery, Colin MacLeod and Martin McCarthy, worked to determine the biochemical nature of transforming principle in Griffith experiments. Okay, so and how they started the working, they tried to, okay, that means they worked to determine the biochemical nature of the transforming principle of the Griffith experiment, and how they continued that experimentation. They purified the biomolecules. They tried to purify the biomolecules just like the protein, DNA and RNA from the heat-killed S's to see which one could transform live R cells to S's. Okay, so that they purified the biochemicals from heat-killed S cells using suitable enzymes. Okay, so this is how they conducted the experiment. And here from the above experiment, what they identified. So they discovered that the digestion, the digestion of the protein and RNA using proteases and RNases. That means the name of the enzymes did not affect transformation. Okay, so that the transformation, how it is happened, the transforming substance was not a protein or RNA. Okay, so and another possibility, the digestion of DNA with the DNase inhibited transformation. It means that DNA causes transformation of R cells to S cells. Are you getting what I am telling? That is, DNA was the transforming principle. Okay, are you getting? See, whatever the experiments, they were conducted, I am telling about the conclusion. They continued the experimentations. So as I was telling about the purification, the purified biomolecules, so that from the heat-killed S cells to see which one could transform the live R cells to S cells. Isn't it? Try to note down. They mixed, that means heat-killed S strands plus proteases plus live R strands. So then they identified the transformation. And the second experiment, they mixed the heat-killed S strands plus RNase plus live R strands. Again they were identified the transformation. In the third case, heat-killed S strands plus DNase plus live R strands. Then they identified no transformation. Okay, so that they were discovered. The conclusion of these, the experiments, in the first case, what they were discovered, the protein of heat-killed S strain is not the genetic material, as there is a transformation of the R strain into S strain in the absence of protein. That was the discovery with respect to the conclusion of the first experiment. And the second one, as I said, heat-killed S strains plus RNase plus live R strains, they identified the transformation, right? Yeah, what happened in the second case, RNA of the heat-killed S strain is not the genetic material, as there is a transformation of R strain into S strain in the absence of RNA. Okay, the last one, the final one. Okay, the final conclusion, whenever they mix the heat-killed S strains with DNase plus live R strains, they were identified no transformation, right? So that the DNA of the heat-killed S strain is the genetic material, because the DNA is get digested with the DNase and mixed with RN, that is R strands, so that enable to transform R strands to S strands. So these are all the conclusions with respect to the, so what is that experiment? Yes, second one, that is the biochemical characterization of transforming principle. Okay, so these are very important two experiments they were conducted. The last one, the third experiment with respect to the search of genetic material. Okay, yeah. So let us discuss the Hershey's experiment. So this is commonly called the Blender's experiment. Okay, so this is conducted by, there are two names of the scientists. The DNA is the genetic material is proved by Alfred Hershey and Martha Chase in 1952. Okay, so that means Hershey Chase They worked on the virus that infects the bacteria called bacteriophage. Okay. So then how they conduct the experiments? Hershey Chase grew some bacteriophage viruses on a medium containing radioactive phosphorus, that is phosphorus 32 radioactive, and some other on medium containing radioactive sulfur, that is sulfur 35. Okay. So and whatever they selected, the viruses they grown in phosphorus 32 got radioactive DNA because only DNA contains the phosphorus, isn't it? When we're discussing about the components of DNA, I told you, isn't it? The phosphorus, that is phosphate group which is present. Do you remember? Yes. And the virus is grown in sulfur 35 got radioactive proteins because the proteins contains sulfur. Are you able to get it? What I'm telling exactly? Because so in the beginning, how they were, started the observation that during the normal infection in the bacteriophage, first attaches the bacterial cell wall and then inserts its genetic material into the bacterial cell. Isn't it? This is the principle why they were selected like this only. Okay, then generally what will happen? The viral genetic material became integral part of bacterial genome and subsequently manufactures more virus particles using host machinery. Okay, so that's what the Hershey and Chase worked to discover whether it was protein or DNA from the viruses that entered the bacteria. Okay, so that's why, the Hershey and Chase, they grew some bacteriophage viruses and a medium containing radioactive phosphorus and some other medium containing radioactive sulfur. Okay, why I'm telling this one? The reason behind. Okay, why he choosing this is only. Okay, fine. So these preparations were used to separately to infect Escherichia coli so that the viruses grown in a radioactive phosphorus have radioactive DNA but not radioactive protein, because phosphorus is present in DNA, not in protein. Okay, as well as the viruses grown in radioactive sulfur have radioactive protein, not radioactive DNA, isn't it? Because the sulfur present in protein but not in a DNA. Okay, then, and these preparations were used to separately to infect the Escherichia coli. When they left to infect, after the infection, they observed that E. coli cells were gently agitated in a blender to remove the virus particles from the bacteria. Okay, and then the culture was centrifuged to separate the lighter viruses particles from the heavier bacterial cells. Here they have given the experimentations. I hope you can identify the medium containing bacteriophage having protein labeled sulfur, that is nothing but yes 35, and another medium containing bacteriophage DNA labeled with phosphorus 32. Okay, this is actually they were conducted experiments in the 3 stages. The first stage is the infection, the second stage is the blending, and the third stage is the centrifugation. Okay, so you can ask me how it will be. Yeah, whenever the bacteria infected with viruses having radioactive DNA were radioactive, so that means that is the DNA had passed from viruses to bacteria. Okay, and the bacteria infected with viruses having radioactive proteins were not radioactive. Okay, so that means the proteins did not enter the bacteria from the viruses. Okay, so what it proves that? So this proves that the DNA is the genetic material. Okay, this proves that the DNA is the genetic material. Okay, so let me to conclude. So what is the main observations in the conclusion of the Hershey's experiment? In the briefly, see, he made an observation that the bacteria infected with viruses that had radioactive DNA were radioactive and no radioactivity in the supernatant. Okay. So, and the bacteria infected with viruses that had radioactive protein were not radioactive, but radioactivity found in the supernatant. So, actually, the main process this takes place in the three-step mechanism: the infection, blending, and centrifugation. If you ask me what is the infection, as I told you in the beginning also, so that is radioactive phages were allowed to attach to the E. coli bacteria. That phages transfer the genetic material into the bacteria. And the second stage, the blending. The viral coats were separated from the bacteria surface by agitating them in a blender. The last stage, the centrifugation. The virus's particles were separated from the bacteria by spinning them in a centrifuge machine. So from that above experiment, what is the conclusion? So that the DNA is the infecting agent that made the bacteria radioactive. Hence, the DNA is the genetic material, not the protein. Okay, let me to conclude once again. The Hershey's experiment are common as the blender experiments. The conclusion of the experiment is the DNA is the infecting agent that made the bacteria radioactive. Hence, DNA is the genetic material, not the protein. Okay, so let it be. So in this class we will discuss about the three important experimentations, because these three experiments are very important for the purpose of to study the genetic material. That is nothing but the search of genetic material. In that one, first one is transforming principle, which is discovered by Frederick Graff, and the second one is biochemical characterization of transforming principle, which was discovered by, there are three names of the scientists: Aswad Avery, Colin MacLeod, and another is McCarthy. Okay, the final one, the Hershey's experiment. It is popularly called Bender experiment. So it is proved by Alfred Hershey and Martha Chase in 1952. Okay, so let it be. So in the next coming classes, we will continue with respect to the properties of genetic material. Thank you. Have a nice day.